Okay, so looking at stele, evolution of stele, okay, in pteridophytes. So what is a stele? A stele is this cylinder, okay, this cylinder which contains, let's say there's a xylem core, okay, in the middle there's xylem. Okay, let me draw this way. Here, this circle, let's say this whole circle is full of xylem cells, okay, and then surrounding this circle is another circle, okay. Then let's say that this circle outside of this xylem core, these are all the phloem cells, okay, and all this is within the epidermis, pericycle, so on and so forth. So the stele means that part, okay, that part which contains the xylem, the phloem, the pericycle, and sometimes the race and the pith is known as the stele. Okay, so let me draw. So in higher plants, you will start with a pith, no, green cells, right? This is the pith. I'll just write P or whatever that is, small p. Then around that, some cells are there. Okay, let me draw like this. Around that, some cells are there which are XY, that is xylem. Then around those, even more cells are there. Okay, so these cells are the phloem. Okay, then sometimes you have medullary rays. Okay, medullary ray cells from which your the leaf veins and all will come. So this structure which contains the xylem, the phloem, the pith, the pericycle, medullary rays, this structure is known as the stele. I hope it's clear. So there it's written there, you can see it. Then these scientists, Van Tighem and Duliat, they developed the stellar theory. Okay, so they use the term stele. Can I zoom it? Okay, I think that's better. They use the term stele in collective sense. If I zoom also, I cannot read the whole sentence. Okay, so they use the term stele in collective sense and mention that the stele is not only made up of xylem and phloem, but other tissues such as the pericycle, the vascular rays, and pith are also associated with it. Okay, so before, in simple terms, people used to say stele is what? Xylem and phloem. Okay, that is what you knew also. But now, this scientists, Duliot and Tighem, they say it contains pericycle also, rays, vascular rays also, and pith also. So according to them, they say that the cortex and stele are the fundamental parts of a shoot, and both these parts are separated from each other by the endodermis. Okay, so if you remember the structure from the, the laginella and the TS, no, you do the TS, you remember outside of the pericycle, this is the pericycle, then just outside is the endodermis. Okay, I think you still remember that. Okay, then let's look at the next sentence. In higher plants like ferns, gymnosperms and angiosperms, leaf traces are large and it plays an important role in the vascular system of the axis. Okay, so let's say this is the stele, means xylem, phloem, pericycle, endodermis. Okay, then there is something known as a leaf trace. Okay, this one, let's say these are group of cells that we say leaf trace, means from this group of cells it will reach the middle vein of the leaf and will give rise to the leaf. Okay, so this is what the sentence means. Okay, now let's look at the next paragraph. So I think this much is done. Now let's look at the types of stele. Okay, so from Jeffrey, okay, so please remember this scientist's name, Jeffrey, 1868. He interpreted the stellar theory from the phylogenetic point of view. Phylogenetic point of view means how they evolve. Okay, let's say you know that man, okay, we came from apes. You say that, right? Man, we came from apes. But actually, we came from Homo sapien. We came from Homo erectus. Homo erectus is when he started to walk on on his 2 legs, okay, almost he started walking on his 2 legs. Then Homo erectus, he came from the Neanderthals who used to walk on 4 legs, okay, we used to walk like animals, no, we used to walk like monkeys on 4 legs. 4 legs means hands and feet, okay. So phylogeny means how we evolve, okay, like we evolved from this primitive one and then we evolved into a more advanced one, no, then from here you got a Neanderthal, then down here you got a Australopithecus. So this is what Jeffrey is trying to explain to you. He's trying to explain how the stele evolved, okay. So when you see this word that he interpreted the stele from the phylogenetic point of view, it means he's explaining to you how it evolved, okay. Okay, so most of the scientists, they see that there are 2 types of stellar organization, that is the Protostele and Siphonostele. So the question used to come like this: Protostele, you have to write about them. Siphonostele, you have to write about them. Okay, so these are the 2 main types. Okay, now let's look at the Protostele and the types. Okay, then a stele where simply this is a xylem core. All these cells are xylem. Okay, all these cells are xylem. Okay, the xylem core, this solid xylem pipe, is surrounded by phloem cells. This is what we are calling the protostele. Very easy. So see, they have written here also, solid core of xylem surrounded by phloem, then pericycle, then endodermis is known as protostele. So very easy. Xylem, then surrounded by phloem, then surrounded by pericycle, then surrounded by endodermis. So this is simply your protostele. Okay. Now haplostele, this one is the simplest kind of protostele. Okay, simply smooth xylem surrounded by phloem is haplostele. Just like I drew just now, like this, no? Yeah, just simply xylem surrounded by phloem is your protostele. So this is the type of haplostele which shows a star shape. Okay. Protostele, if I show you, let me show you from here. Here, this one, this is the protostele. You can see, no, the xylem is right in the middle. Then there's a metaxylem. Metaxylem, these are the older xylem. Protoxylem is the younger ones, means the smaller inside xylem. Okay, the black color is the protoxylem, means the smaller cells of xylem. When they grow up, they become bigger and they are known as metaxylem. Okay, then this whole xylem, proto and metaxylem, is surrounded by the phloem. Okay, here this whole xylem, both the proto and the metaxylem, is surrounded by your phloem. Okay, here protoxylem, these are the young ones. Okay, the cells, they start very small, no, and young. Then as they become bigger and larger, no, those xylem are known as metaxylem. Okay, so this is clear, I think, from the image. Protoxylem. So let's go back up. I'm sorry if some of you feel like vomiting when I go it too fast. Now if you look at actinostele, actin from the word star, okay, means when the xylem core has some radiating ribs which give it a star shape. From here you can see the star shape, no, the star shape is very clear. Okay, I'll draw here the plain one. This one like a star. Okay, this is your xylem, then surrounding it all around is your phloem. So this is your actinostele. Okay, actin from the word star. You know, actin is star, no? Okay, so this one we have to look at some example. Example from the word star itself, asteroxylon. Okay, so from the word star, asteroxylon. Okay, in protostele, the example was, in haplostele, example was, you can give in Selaginella crossiana. Okay, this one you know, you've already known this one. So you give this example, Selaginella crossiana. Okay, now looking at plectostele, plecto means plates. Do you see here parallel plates? Okay, so when the xylem appears like parallel plates, and in between the plates you have the phloem. Okay, you can see the phloem in between the plates. This is your Protostele. Alright, example was Lycopodium volubile. Now if you look at mixed Protostele, here mixed Protostele, in this image you can see that there are some Protostele, means some xylem cores in between, okay, in between the phloem. So this is your mixed Protostele. Now again girls, you have to draw these diagrams, that's how you will understand them, okay. But let me tell you that you don't have to draw, if you draw these diagrams, these in between, then you don't have to draw the, what I'm going to say, I would prefer if you just wrote down all the, the words, and then you just draw this big chart, this one, okay, which shows how they evolve, okay. So you see these arrows? From this Protostele, okay, you get the actinostele and the ectofloic siphonostele, then you get the amphinoic, they're giving you arrows showing you how they evolve, okay. I would prefer if you stick only with this diagram, okay. The rest of these diagrams in between, you should not draw because it will spend your time, okay. These diagrams are actually just for your understanding, okay. So write only the wordings and draw diagram, you give only this big one, okay, this big chart. So I hope you have understood that much, okay. These diagrams in between are all for your understanding, okay, because when you look at the diagram, it's so much more clearer. Okay, then again, if we look at this siphonostele, there are some means a medullated protostele is called a siphonostele, means they will have medullary rays, means there are some steles here, and there are some gaps in between, okay. When these gaps are there, these are known as your siphonostele, means these medullary rays are there in between. So let's look at how they originate. So a stele having a tubular vascular region and central parenchymatous region was evolved from the Protostele. So again, the siphonostele, it came from the Protostele itself. Then the pith is surrounded by xylem in the siphonostele. So what is different from the protoxylem? Okay, do you remember? If I will draw here, if here is the protoxylem and this is the siphonostele, remember the protostele, xylem directly, then phloem, then your endodermis and your pericycle, whatever. Okay, but in siphonostele, you don't start directly with xylem, you start with pith. Okay, I'll just write here PI for pith. Then you have your xylem, this one ZY. Then you have your phloem, this is your pH. Then your endodermis and pericycle. So what's different here is that in siphonostele, you will have one pith. Very good. Okay, so let's go on. So there are two theories about how the pith originated. Some say it came from the interstellar region, means from the middle. Okay, means from the middle. Some cells, they just started behaving like a pith. Okay, then some are saying it's an extrastellar origin, means it's not the cells of the middle that behave like a pith, it's the cells from the outside. Okay, so if this is the xylem and phloem, means these cells from the outside, they started piercing inside, traveling on the inside, and they join the mass in the middle and become the pith. Okay, so interstellar region means these cells of the phloem inside, of the xylem inside, they just started behaving like a pith. Okay, then the xylem was left only on the outside. Okay, so that's your interstellar. And extrastellar means the cells from the outside, they just came from the outside towards inner part to form the pith. Okay, so I think it's clear. Now let's look at the types of siphonostele. You have your ectofloic and your amphifloic. Then simply let us look at the images again. Okay, instead of learning by heart, just look at the images. So in ectofloic siphonostele, you are seeing what? You saw the pith. Okay, you're seeing the pith. Then you're seeing the xylem, then you're seeing the phloem. Okay, then endodermis, pericycle, and then a leaf trace. Now let's look at the amphifloic. Now, do you see something different here? Yes. you are seeing that there is a pit first, okay, as usual, Siphon Steely, but then instead of xylem, we are getting phloem from the inside, okay. Then in the middle you're having xylem, okay, and then outside also you're getting phloem, okay. So what is the difference between amphiploic and ectoploic? If I draw in the side, means pit, xylem, phloem, this is your ectoploic. If you have pit, then phloem, then xylem, then again phloem, this is your amphiploic Siphon Steely. So just from the diagram, you have already understood. Okay. Again, now looking at the Stele. Stele is when ectoploic Stele and amphiploic Stele is the same as the previous. The only difference being there is a, you see some gap here, there is a leaf trace, okay, and a gap, okay. So it's exactly the same. Here ectoploic is where you have pit, then xylem, and then phloem, okay. Then here amphiploic you have pit, right, you have pit, then you have phloem, okay, then xylem, then phloem again, okay. The only difference in Stele is that you have this gap, okay, you have this gap and the leaf trace, okay. So it's very easy, this from the diagram itself. Now these are the different types of Polystyly, where you have steles in small circles, okay. So I think from here, from the diagram, it will be very much clear, okay. So if you draw just this main diagram, it's more than enough, all right. Instead of drawing all these diagrams are meant for your understanding. So you write just the text, okay. I hope you have understood. Write just the text part and then the big diagram, this big diagram showing the evolution as a whole, okay. Don't draw all those other diagrams, okay. Thank you.